Okay. Welcome back, everyone, to the afternoon session. Isn't this a fantastic meeting? I mean, really, this is amazing. So thank you to the organizers, of course. Of course, it's a great honor to be chairing this session, especially as the token crystallographer, who I've learned this morning that I won't have a job soon anyway, so it's the last time you're going to see me. I don't think that's true, just to be clear. So we've heard about the amazing potential of Cryo-EM in the next 50 years. And we've also seen the rapid growth of single-particle cryo-EM over the last decade. But really one of the most exciting promises of the technique is to be able to routinely look at biology in situ. I mean, that really is going to be transformational. And so in this session, we're going to hear about imaging molecules in the native environment, accelerating the technology for Cryo-ET, and for being able to look at molecules and say where they are in the cell. So our first speaker is Nigel Unwin, who I'm sure you all know anyway, but just to say, he spent a career focused on, I think, challenging membrane protein biology using electron microscopy techniques, notably bacteriorhodopsin with Richard Henderson, and a lot of time spent on the nicotinic acetylcholine receptor. And so he's going to talk about Cryo-EM of that system in the native environment. So thank you, Nigel.
Thank you, Paul, and thank you, Bob and Eva and Holger and Wire. It's an honor to be here and, of course, a great pleasure. Before I start talking about receptors and membranes, I thought I'd take a moment to recount my early connections with Berkeley and actually my involvement with Cryo-EM, which was in the 1980s, and so that's when Cryo-EM sort of started. So actually, I began my research at the lab where I am now, the Laboratory of Molecular Biology in Cambridge, and in the late 1970s, I wanted to work in California. So I ended up working at Stanford for 7 years. And when I was there, of course, Stanford's quite close to Berkeley, and there was quite a lot of opportunities to interact. And I went to Stanford, and actually we set up group meetings, Bob and me. Where's Bob? I don't know if Bob remembers this, but they were a lot of fun. And I thought I'd show you the people who were around at Stanford at the time. Actually, Werner Kuhlbrand shouldn't be in that picture because we left him at Cambridge to finish his PhD. And Andres Engel was only there for a short time. But the other ones were there for quite a while. Ron Milligan, Ed Gogol, Mark Yeager, Paula Flicker, Chris Aki, of course, Liz Kubalek, Chikashi Toyoshima, and Alain Brisson. So we were all sort of starting Cryo-EM then. Of course, Ken Taylor had moved on, but actually he was at the laboratory in Cambridge before I left, and we interacted quite nicely. And one of the things that Ken did there was build a cold stage. I encouraged him in the workshops, built it, and then I took it to Stanford with me. And this actually led us to get the first structure gap junction channels from crystalline sheets of gap junctions. You can argue whether that's a proper Cryo-EM structure in ice because the temperature of Ken's stage only went down to minus 120 degrees, which is a bit warm. But Bob and Ken sort of helped us out by they developed a method where you sort of squeeze, it's called the carbon sandwich method. I don't know if anybody knows about that, but you sort of squeeze your specimen between two carbon films. And the thing was that when you did that, there's not much water around, and I think there was too little water there for the ice to crystallize. So it was proper Cryo-EM, even though it was at minus 120 with amorphous ice. But actually, at the same time, we were developing the technology based on what Jacques DeRosier had done with the rapid freezing, and got proper Cryo-EM pictures, this time using a Gatan cryo holder that Peter Swan at Gatan had helped us to develop. Well, after that, I went back to Cambridge, and I had some good collaborations with Chikashi Toyoshima, and actually an extensive time with Yoshi Fujiyoshi and Atsuo Miyazawa in Japan. But this meeting is about the future, so I'm going straight to the introduction of the future by talking about the present, which is, of course, using detectors. Where in between what I've talked about and now, there's thousands of films have gone down the drain. But now it's easier in many respects, and of course you get these nice pictures. Now, the acetylcholine receptors and torpedo postsynaptic membrane. To explain first of all, the postsynaptic membrane is a specialized part of the electrocyte cell in the case of torpedo, and or the muscle cell, because the two cells are very closely related, where the nerve terminals are butt against the muscle or torpedo cell membrane. And there they form synaptic junctions. They release acetylcholine, the neurotransmitter, and transiently, and it binds to the acetylcholine receptors, which are heteropentameric ion channels in the membrane of the torpedo or muscle cell. They open a small hole, the membrane depolarizes, and that's a signal for generating muscle contraction or an electric shock in the case of the torpedo. In the cell, there are two organizations of receptor. One is very dense organization at the synapse, and the other is a more random organization. And when you isolate the vesicles, you destroy that, but if you leave them around for a while, they sort of segregate into their prior organization, and that some of the receptors get really close packed. So close packed, they're crystalline order, and they form these membrane tubes. And other receptors remain randomly distributed in the membrane. It turns out, well, at the synapse, they're very densely packed, and it turns out that the organization, this is a freeze fracture picture, in situ freeze fracture picture from John Heuser, showing the organization at the synapse. And you see that the receptors form ribbons of dimers. Well, it's the same ribbons of dimers that are formed here in the tube. Same membranes, same organization of the protein. And in muscle, ribbons of dimers as well. So that's the functionally interesting organization that is relevant to synaptic transmission. Now, it turns out that people, of course, have isolated the protein, looked at it in nanodiscs and detergent, and mainly Ryan Hibbs' group and John Basinger's group, and they get beautiful 2 to 3 angstrom resolution pictures of the channels. Well, in the membrane, the channels have a different structure. The helices going through, the transmembrane helices, these vertical ones, are more splayed out in the membrane, and this helix that lies on the bottom membrane surface is flat against the membrane surface. And to understand the difference, one needs to understand how the lipids are organized in the membrane a bit. So I'm now going to tell you about how the lipids are organized in the synaptic membrane as we see it in torpedo. And we're looking at a helical reconstruction from these tubes. And what you notice is the high contrast features, which are the protein, and these two grey bands spaced 30 angstroms apart, which arise from the phospholipid head groups of the lipids. Now, these membranes have a lot of cholesterol, at least 40 mole percent. And of course, you can't see it in these pictures directly because it's sort of intermingled with the phospholipid head groups, which are dense. And the cholesterol only has a hydroxyl, so it would produce a blank where the phospholipids are. But in some cases where it's concentrated, like here, you see gaps between the phospholipid head groups and the surface of the protein. That's for sure telling you that there's a concentration of cholesterol in the membrane there. One interesting thing about the membrane, when you look at it with the protein masked out, is that it's asymmetric in the density distribution. The density trough in the middle of the membrane is well, it's not exactly in the middle of the membrane. This bottom peak representing the intracellular phospholipid head groups is about 3 angstroms further away from the low density trough than it is there. And what that means is, from people's studies of mixtures of cholesterol and phospholipids, is that the cholesterol has ordered the fatty acid tails of the phospholipids, making them straighter so they extend a bit. That's how the change in thickness arises. And you can estimate that from that shift there, that there's about twice as much cholesterol in that leaflet as there is in the other leaflet. And so that means it must be well over 50 mole percent, which is the saturation amount of cholesterol you can have in membranes before it starts precipitating out. So in the bottom intracellular membrane, the cholesterol monomer is no longer stable. So I just want to show you now sections in the plane of the membrane, looking at the extracellular leaflet there and the intracellular leaflet here. And sorry, the contrast has been reversed because the hair slices through the helices that are going through the membrane, making the receptor. Then outside that, the grey area is the phospholipid head groups. And then close to some parts of the protein surfaces, you see these black regions, the same density as the hole in the center of the channel, which must represent accumulations of cholesterol. So it binds on the subunit surfaces in specific places, and also connecting the delta subunits of neighboring receptors. And this, by the way, is a ribbon of dimers of the receptors that I mentioned earlier on. Now, the phospholipid head groups, you can tell they're disordered there. There's no sort of recurring motif, and they're probably fluid. When you come to look at the intracellular leaflet where the cholesterol is high, the phospholipid head groups are just above the helices there is grey. And then you go slightly further into the hydrophobic core, and you see these lines of density, which are spaced about 6.2 angstroms apart, which is, that's highly significant because I made some monolayer crystals of cholesterol, and they form linear arrays that are 6.2 angstroms apart. So these linear arrays must be some cholesterol-driven assembly, a mixture probably of cholesterol and the initial saturated portions of the fatty acid chains, which have about the same cross-section actually as cholesterol, can fit in the same sort of lattice. And we sort of know that this is right too, because when you look at sections through the three-dimensional maps, the thickness of these striations is almost 9 angstroms, which is the same as the length of a sterile group. So what we see lying above those transmembrane MX helices is the sterile occupied portion of the bilayer. Now, I want to tell you about this MX helix lying against the sterile occupied portion and the pore there, which has a tapered shape. And so we only have a resolution of we claim 4.7 angstroms, and this is an atomic model from a nanodisc structure fitted to the densities. And yes, you see the helices lying flat, all of them lying flat against the membrane surface. And the other important feature is when you do a surface representation, this is you can see the purple is the polar groups which are facing the phospholipid head groups, and then there's the rest of it's hydrophobic. But you see a very flat surface as though it's engaging with the cholesterol-ordered lipids. In the cholesterol monolayer, the hydroxyl of the cholesterols are actually atomically flat, so you two almost perfectly flat surfaces there, so they're engaging with each other. Then if you look at the pore, it has this characteristic tapered shape, and I've emphasized leucine residues in the middle there, which have been shown by biochemical experiments on membrane vesicles to function as the gate of the channel. They form a hydrophobic ring, and the hydrated ion, there's quite a hole down the middle, but the hydrated ion, it's about 8 angstroms in diameter and can't go through, so that's why it functions as a gate. When you look at the nanodisc structure, it looks like that. Now you can see it's quite different. See, instead of the helix, the pore being tapered, it all comes in contracted. And actually the narrowest part of the gate there is right at the top, leucine 16 prime. So we have two structures of the channel. One is in the normal natural membrane, the other is in nanodiscs, an artificial lipid environment. And how one gets from one to the other, you can see by superimposing the two structures and looking at the regions where they change the most. So blue is the nanodisc structure, and orange is the membrane structure. And of course, blue has the tilted MX helix, as I mentioned in the beginning of the talk. And you can see when you compare these two, when you untilt MX to make it match up with the proper membrane structure, it brings the transmembrane helices with it, in particular M4, the outside helix. And this is easy to see when you're looking at the structure because there's this really extensive hydrophobic contacts between MX and M4. This happens in the delta subunit, and the gamma subunit does the same sort of thing. So these two helices splay. This pushing it down flat makes the helices splay out. And then the other ones sort of move by rigid body movements. If you look at the alpha gamma subunit here, you can see all 4 helices just sort of shifted out by about an angstrom. Of course, these changes make the pores totally different. In the membrane, it's a wider pore, but still narrow enough to stop the ions going through. And in the nanodisc, it's a very contracted pore. If you compare it with a muscle structure, the muscle structure is more tilted, and the pore is more closed. So we have two interconvertible forms of the receptor. One is in the cholesterol-rich lipid environment of the synaptic junction, and the other is in the artificial environment of the nanodisc or in detergent in the muscle case. So the big question is, do both forms exist in a natural setting? Well, they could do, because we see two sort of distributions of the receptor. Like this, there's the ordered distribution, where we see the receptor in the membrane. We know that's what's happening at the synapse. But this part at the bottom here is a disordered membrane, and it's possible that the nanodisc or the detergent ones resemble these receptors here. We don't actually know that. We haven't done the experiment yet. But just before getting on to the final bit, I want to just emphasize that the difference between these membranes. This one is the membrane actually in the intracellular leaflet. It's not even, the lipids aren't mobile, otherwise you couldn't take an image with or do the image processing. That shows the striated appearance of the cholesterol driven assembly. But this one is disordered and fluid, and there's two ways I know about that. That is because, first of all, you can do 2D class averages and show very convincingly that the thickness of the bilayer, the head group, the peaks of the head groups are separated by less than 30 angstroms, like they are in the ordered membrane, and that suggests that there's less cholesterol, so that the lipids can flop around more and make the membrane thinner. And the other thing is that part of the membrane is quite fluid, because you can make use of the fact that the receptors don't like being in regions where there's negative curvature, or flat regions like that. So what you can do is take a round vesicle and just flatten it, and then you find the receptors sort of move over to the side. So translating this to nature, we may have two sorts of receptors here: junctional receptors, ones at the synaptic junction, and extrajunctional receptors, which may be like the nanodisc or detergent ones. The junctional ones are the ones with a tapered pore and the flat MX helices against the membrane, and the other ones have a contracted pore and the helices are tilted. And actually, the main difference, if you look at it like this, straight down the channel, is that in the membrane, the interfaces between the subunits are really quite wide apart. And in the nanodisc or in the detergent, they're quite close together. They come much more contracted. Whoops. Well, what I want to say is that in the one at the synapse, then the hole down the center is wider, and the interfaces are wider, and that would suggest, to me at any rate, that maybe when it opens to a bigger hole through the membrane, and actually opens more rapidly because the interfaces are weaker. And actually it turns out that almost 50 years ago, electrophysiologists working with denervated and normal nerve muscle fibers showed that there are exactly two forms of receptor, one at the synapse that has a high conductance and works 2 or 3 times faster than the ones that are not extrajunctional outside. So that's a good hypothesis based on the data that we've got at the moment. But of course, not sure that the nanodisc structure is actually what they represent. So my final thing to say is that for the next 50 years is lipid context matters, not only just seeing the thing in the membrane, but whereabouts in the cell membranes the protein is. So one needs to know if the structure of a solubilized protein recapitulates accurately the structure it has in the membrane in which it functions. So I think that validation by in situ cryoimaging will become an important step. I mean, we have validation of structures by Fourier shell correlation and all that, but I think, you know, it'd be good if people sort of go one step further and show when you publish papers that it is actually something that exists in the cell. And the other thing thing that I'm sort of particularly interested in is that I think that this ordered structure that you see in the membrane, people don't look at membranes yet, but I think it'll be quite a common feature because plasma membranes are sort of 30 to 40 mole% cholesterol, and you only need a little bit of asymmetry and you reach saturation, and then the cholesterol and fatty acid chain should precipitate out. And you notice this is a pure two-component system here. It avoids all the heterogeneity of the head groups and the heterogeneity of the tail groups. And so I don't see why in lots of plasma membranes you don't get this sort of organization. And maybe this is the sort of underlying structure that exists in what people call lipid rafts. So thank you. I want to say thank you particularly to Bob and Ken, who sort of got us going, you know, through talks at when we visited Berkeley, and thank you all for listening.
Thank you, Nigel. That was fantastic. Questions?
Hi, great talk. So the cholesterol flip-flop rate in bilayers is on the order of sort of micro to milliseconds. And so your observations of kind of preferential localization of cholesterol in the inner leaflet are kind of suggestive that that is specifically bound cholesterol rather than freely accessible cholesterol. I'm wondering whether you can test this experimentally by, for example, freezing in the presence of different concentrations of beta-mercaptoethanol or something like that.
No, this is not cholesterol that's touching any protein surface, of course, because it extends across quite a wide space.
But then it should freely flip-flop, whereas your observations that it's preferentially localized within one layer suggest that it's probably specifically bound to that protein.
Not necessarily. We don't understand enough about membranes. What makes more cholesterol be in one membrane than the other, one leaflet in preference to the other. I mean,
yeah, it would be cool to test. I think, yeah, it's a nice observation, and it would be cool to test using, you know, cholesterol assays, etc.
Yeah,
Thank you.
You could test.
Nigel, so it is related to this because the lipid composition and the curvature are linked, and I just don't know what is the cause or what is the effect. So in the tube, you obviously has one direction of very high curvature, where you would, you know, the lipids with the smaller polar head groups will go inside. So I don't know whether that arrangement is what is there, and that's what stabilizes the tube. So whether
no,
it's
you know, if you look at the neuromuscular junction or the torpedo synapse, the receptors are concentrated on the curved surfaces, and when you go to a flat surface or one that curves the wrong way, you don't see receptors, or very few of them. So it's absolutely natural in that regard.
Right, but just in response to, you know, there's some kind of linkage between...
Yeah, that's right. That might be one way of preferential getting the cholesterol on one side because of the shape of the cholesterol and everything else.
Any other questions? Okay, great. Let's thank Nigel again. Okay. So as Bridget's getting set up, we're going to now move on to thinking about technology to try and make cryo-electron tomography a bit more automated. So our next speaker is Bridget Carragher, who's known worldwide for her efforts to try to automate single-particle cryo-EM. And she's going to talk today about what we can do for electron tomography. Look forward to this.
Thanks very much. I too am very honored to have been invited today, and I'm honored to be representing the Imaging Institute. There's many of the members of that Imaging Institute here, and I'm simply representing them. So for those of you who don't know, the Imaging Institute is a new institute. It was set up about a couple of years ago. And our mission is to enable deep insights into the architecture of complex biological systems at the molecular scale. That's a very big mission, and we intend to carry it out by pursuing a number of grand challenges. And the first grand challenge that has been defined is to develop technologies and methodologies to image the molecular architecture of the cell at atomic resolution. And as we all know, right now, the only game in town for that is cryo-electron tomography. So you'll hear a lot more about cryo-electron tomography as the day goes on. Suffice it to say, it's an exceptionally challenging and complex pipeline at the moment. There are bottlenecks and challenges at every step. We need labeling techniques. We need super resolution correlative light microscopy. We need to cut these exquisitely thin sections. There's all kinds of problems and issues with doing that. We need to acquire a whole lot of tomography data, maybe 2D, maybe 3D, we don't know yet. And then we need to process it by very complex processing pipelines. We have to annotate it, really hard to do, and then finally do subtomo averaging in order to get these high-resolution structures out of the in situ structures. So this requires technology development at every single step that is better, faster, smarter, and improves the whole process. And we have a whole bunch of sort of requirements that we've set up and metrics that we can measure for how we will improve this and how we can keep ourselves honest in measuring that improvement. But suffice it to say, we developed a roadmap at the Institute, and it consists of building really high-end technology platforms, pursuing a whole bunch of technology research and development projects, and driving them very tightly by biological drivers. We have never believed in developing technology in a vacuum. You don't develop anything useful unless you stay very, very close to the biology. So the technology research and development is many, many different projects, and today I'll just highlight a couple of them given the time we have. And the very first one, and the very large one that we're pursuing at the moment, as you heard from Holger earlier, is to try and help out developing a dual laser phase plate. So Holger told you all about this, and Holger's whole team should be up here, but he's representing that team. And that's what they've been doing for many, many years. And our job at the Imaging Institute is to take that technology, miniaturize it, simplify it, make it robust, and squash it into a cryos box, and then make it easy and robust to use. So that's what we've taken on. It's a very complicated collaboration between, hugely Holger's group, as well as Thermo Fisher and an engineering company called Applied Precision Design, which has been doing the design and development for us. And of course, it would be remiss not to mention Bob, who has been absolutely pivotal in driving this project right from the very get-go for more than a decade now. So the first thing we needed is a microscope to put it in. You heard about Holger's microscope. We have a slightly different design. It has a unique transfer lens. And as a result of that, the performance is quite good. Good enough we don't need a CS corrector, which is helpful, because it's very tall already with that transfer lens. The specimen's up here, and the laser phase plate goes in there. The interesting thing about getting this unique microscope was that because they gave us a unique design, it got delivered a year early, and that never happens in real life. So that was quite a surprise. So the microscope's there. We have been developing this dual laser phase plate. What that means is two crossed lasers. So this is the thing that is going into the microscope next week, and this is the relative size of it. It's a tiny little device with all kinds of exquisite controls, piezos and bellows and all kinds of fancy little micro machined systems to align the system and keep it stable. And then, of course, there's these fancy mirrors. Now there's 4 of them crossed in the device. And Holger mentioned that there may be some advantages to having this dual device. Where are we right now? As of today, That whole system has been built out in a clean room on optical boards. We have a device that's in the microscope now to align the laser faceplate and guide it in. And on Monday, all of that will be taken down and transferred to the Krios room and packed inside the microscope. And then what we hope to do is spend the next quarter characterizing, testing and integrating that laser faceplate. In the second quarter, we'll optimize data collection. And by the end of the year, we hope to have a device that anybody can come and collect data on and is robust and useful. And we can start to see how far can we drive the improvements that this laser faceplate gives us to contribute to biology. So that's where we are with that. Monday, it goes in. Wish us all luck. It's a very complicated system. It can go wrong at many, many places. So just to talk about a different technology project, and that's thinking about the Cryo-ET processing pipeline. And by that we mean, the set of software tools that are necessary to take this wiggling, jiggling set of tilted data into a nicely interpreted sociology of the cell, as Wolfgang coined it. So right now, that process can take many months and sometimes even years. Some of the data that is appearing in papers now was collected many years ago. It's complex, it's difficult, there are many different packages, they're all using different metadata, and the whole thing needs to be better, faster, simpler, smarter. So at the Imaging Institute, we think of that pipeline as 4 discrete steps. The first step is going from the raw frames to a nicely best aligned, corrected 3D volume. After that, annotation is a massive step, and then we do subtomo averaging, and then we do atomic interpretation. So for the first step is where we've made the most progress so far. Thanks for Sean Zhang and his group, that's right. He has been developing the software called RTOMO, and it takes the unaligned tilts to the 3D map in a single step without any supervision at this point. And he's also improved it vastly. I won't go through this long list of improvements, but obviously everything about doing this has to be improved to get as good a 3D map as we possibly can. So he's made tons of improvements. That software has also been released to the open source, thanks to CZII taking it over. And it was released last 2023, and Sean does many, many releases. The latest one was just done last week. So along with all these improvements, it was greatly speeded up. Before these speed ups, it used to take people about 4 weeks to process 100 tomograms, going through all these endless different steps that are entailed in this list. Currently, we can do 300 tomograms in 24 hours. And that's about how long it takes us to collect 300 tomograms. So we think of this as real time, because if we went faster on the microscope, we got more, we would just add GPUs and we could keep up. So we're at real time at this stage. What does that mean? It's about 80 times faster than it used to be. But what does that mean in practice? Is that now, instead of collecting hundreds of tomograms, we can collect thousands of tomograms. And that's very important to our biological driver projects. We had a lovely visit with Richard and Chris yesterday, and we were all agonizing over how difficult it is to do tomography. One of the reasons is for single particle, we collect millions of particles and sort them out. In tomography, we're lucky if we have thousands or tens of thousands, but we're certainly going to need tens of thousands of tomograms in order to pull these particles out. So we need that throughput as high as possible. We need it to be as simple as possible. And the difference is feasibility versus infeasibility. For this one project, we have 16,000 tomograms. If you were doing that the old-fashioned way, it would have taken you 8 years to process it. Doesn't mean you would have done that, means you would never have done that. Whereas right now, that presents about a month of work. Of course, there's other things that get in the way, but that's the technology. I just want to give a shout out to Bob here, because Bob has been a big advocate of throughput and high throughput all his life. Clint and I had an interaction with him in about 1999 where we were providing supercomputer resources, and Bob's process was to carry out massively large computations that are needed to merge image data from as many as 100,000 particles, which was visionary at the time, and now we do millions of particles. But that was a very long time ago, and it was very, very difficult to do. And I just want to say that last week, he sent email to a bunch of us saying, I suggest it's time to make quantum computing a topic that is openly discussed. So Bob is always thinking 5, 10, 20 years ahead. The other thing we've done in this pipeline is add denoising. This is just a weighted back projection tomogram that comes out of our tomo, and that's what happens to it if you do these noise-to-noise denoisings using machine learning algorithms. And you can see the sort of night and day difference in what you can see on these membrane proteins after the denoising. You can see a shape there. You know, if we could just do a little better, maybe with a laser phase plate, maybe something else, we could discern that shape and we could start to see what are those proteins in our in situ structures. So we measure everything. We try to measure everything at the Image Institute. The contrast is about 5x improved by the denoising. We've sped the denoising up about 20 times. So this also happens in real time. So while you're collecting your 300 tomograms a day at the microscope, they are appearing as nicely aligned denoised 3D volumes, and that helps a lot. So that's the good news. But the second step of the pipeline is the very bad news, and it's a major bottleneck. As anyone who works in this field knows, that annotation step currently takes months to years. You can get your tomograms, 16,000 tomograms we have, but annotating them is a real nightmare. It's by far the major bottleneck, and that's really why instead of these lovely, rich interpretations of the cell, what we have are membranes and ribosomes, because those are the easy ones to annotate and the ones that can be done. You'll hear probably later this afternoon why annotation is so difficult, but for many reasons. The membrane and particle locations, you need tens of thousands of them to do averaging. The challenge is that the cells are very crowded. The tomograms have very poor contrast. They're not isotropic. The individual particles are sparse and in that very noisy, crowded environment. So the current status is that often this annotation is being done manually, like we used to do in the bad old days of single particle, where people pick particles one at a time. You can't pick 1 million particles. You can't really pick 10,000 without going crazy, and you certainly don't want to do it every day. So we need to automate this. And template matching is a great method, and I think will be the future. Right now, it works so-so. We don't get it for much other than ribosomes. We actually think right now machine learning is very promising, because as people are going to the trouble of manually annotating, we can take that and learn and use it to train machine learning algorithms. But as we all know, one of the biggest problems with machine learning is it needs a lot of training data. And so very early at the Imaging Institute, we set up a Cryo-ET data portal to provide annotated tomograms to the community. And what we really hope to do is engage the machine learning community in this topic. And in the end, we also hope to provide structural data for a virtual cell model, but that's a little bit further down. Where are we right now? We have about 17,000 tomograms and about 30,000 or so annotations. So it's good enough to start doing training, and we've the membrane segmentation algorithms, several of those have been run, 3 or 4 of them now, over the entire 17,000 tomograms, and we can start to do comparisons and maybe consensus on what are those annotations. We also have set up a working group with EMBL, EBI to try and define metadata for cryo-tomograms so we can exchange data across different communities and across software. But what we really want to do is engage the machine learning community. And so what we want to set up is a challenge to have the machine learning community try and do this annotation for us. Oh, I should point out, if you hit on browse data, you get all kinds of nice tools, web tools, etc., where you can browse through the data and browse through the annotations or take it into something like Napari. So what we want to do is set up this machine learning challenge, and the problem with the challenge is you need ground truth. So the first thing we had to design is a nice data set to define ground truth, and we call this our phantom data set. And what it consists of is a lysate where we pull lysosomes down onto a grid. We add 6 other proteins in known aliquots, and then we image that sort of mixture of proteins. And the good thing about this particular method we're using is that the proteins are well distributed across the thickness of the ice, and the ice is roughly 150 to 250 nanometers thick, which is what most tomograms are. So we did that, but to establish ground truth took 3 months of miserable curation by many, many people in the group. We did it across about 500 tomograms, and we annotated tens of thousands of complexes to try and establish as good a ground truth we can. And with that in hand, we launched a challenge using this web-based platform called Kaggle. I had never heard of it. The machine learning community is very familiar with it. And what they do is set up the challenge so that they give people training data, and then they have private sequestered data against which these attempts at solving this problem are scored. And within a few days of launching the challenge, we had thousands of entrants, and right now we have over 6,000. This was 3 days ago. There was 5,800. Now it's over 6,000 this morning. So there's a lot of interest in this topic. And it's a fabulous community. There's a hugely active discussion. There's comments. There's advice from people going back and forth. These people are all contributing notebooks, computer code notebooks, to help each other to try and solve this problem. And they submit new entries every day. So as soon as you have an idea, you can submit it against the data and get yourself scored. And this, was a couple of days ago. Every one of the top 10 people, so you can see how you're scoring, had submitted a new entry within the last 24 hours. So they're hammering away on this even 2 months into the challenge with about a month to go. And I should say they're doing very well. Their scores, 0.776, our very best score using DeepFind-ET before we set up the challenge is way below that. And in fact, we're now in position 418 of these many hundreds of entries. And there's prizes for the top 10. So we're excited about that. We already regard it as a success because as a result of that, we have now this phantom data set that we will make available on the portal, and maybe it'll be a good standard for people to use. We have synthetic data based on that phantom. We developed a whole lot of different software packages in order to do the ground truth, and those are now all being used in our real-world data. And then we're going to have a challenge workshop and hopefully publish the results of that challenge on April 2 to 4. Let us know if you're interested in attending. We'll come after some of you anyway. We'll invite the top 10 winning entries. I should say the top hundreds and hundreds of people in this challenge right now, all 100% machine learning. They had never heard of Cryo-ET, as far as I can tell, until they entered this challenge. So we will also invite members of the community who are doing Cryo-ET picking. And we'll discuss the lessons learned and how to set up future challenges, and most of all, incorporate the very best algorithms into the pipelines and make them available to the public. So we hope to address this annotation problem. I also want to point out Bob has been here and there and everywhere from the beginning. Years and years ago, we set up a similar challenge for single particle data selection, and we published a paper on the results of that challenge. And you can see Bob there as the third author. He was very, very involved in that challenge. And that data set, too, was used for many, many, many years after we set it up, and for people to test new algorithms for particle picking. And finally, I just wanted to mention, we don't do this in a vacuum. We take our biological driving projects very seriously, but I don't have time to talk about them. But members of our group are here if you want to talk to them. And we have basically 3 areas we're looking at: structure of organelles, lysosomes, mitochondria, the structure of cilia and the ciliary synapse, and then the structure of the neuronal synapse. And all 3 of those projects are in active development at the Institute, and we collect lots and lots of tomograms for them, and we make those tomograms available on the web portal, on the data portal, as soon as we can. So anybody who has an interest in these topics may go and take that data and analyze it if they wish. My throat just dried out. Thank you very much for your attention. This is our amazing group. We're just down the road at Redwood Shores, and please come and visit us anytime. Thanks.
Thank you, Bridget. It's great to see things happening so quickly. Questions?
I've got to say, it never feels quick when you're in it. It just feels really slow.
Sure.
Yeah. But it's nice to hear other people see it that way.
I think Eva has a question.
Bridget, it's always so cool, the things that you guys are doing, and I agree with Paul. From far, it seems like you guys are shooting very far and fast. I was just wondering if you could say a little bit more about how, those 3 months of annotations by people and establishing that ground truth. Say a little bit more about that, because everything is going to be matched against that, I suppose, and to me, it's not a trivial thing how that is done, and you would have, even humans will have false positives and negatives that you have to.
100%.
Yeah. I don't know. What was the approach?
Right.
that you guys have used, or were there several approaches and it was like
Exactly.
overlap of
Yeah.
what?
Endless approaches. It's all written up in this paper, which is on bioRxiv, but we use many approaches. And the issue was, what is ground truth? It doesn't exist. People are terrible at this. And in fact, early on, we said, let's have a pickathon. You know, we'll have 35 people or 40 people we had. They came and spent a week at the Institute all picking. At the end of that, we threw all of their picks away because they were terrible. And we had a few experts do it. It's not a crowdsourceable thing at this point, you know, for almost everything. Even ribosomes are very hard to pick. So instead, we had some experts in the group who were really great at it, pick some particles to start. We then trained machine learning algorithms with that, used those algorithms to pick things. Then we did 2D classes, 3D classes, did machine learning to exclude particles. Months and months of iteration, round and round. And in the end, many of those particles were actually looked at one by eye, because we would never do that in the real world, because if you have a few false positives, a few false negatives, who cares? You wash them out, you don't care, you get them with the 2D class averages. But because it was a challenge, we felt we had to get as close to as possible to that real ground truth. So that's what we did. 3 months of work. But now these people are scoring quite well, and they only get 12 hours of GPU time on a pretty normal box. So if they can do in 12 hours what took us 3 months, we will be thrilled. You know? We haven't seen what the models look like yet because it's still sequestered, but the scores look extremely encouraging. It's only lysate, though. We really want to do this on in situ cellular material, and that will be one of the points of this workshop to discuss how do we do that? Because establishing ground truth on this lysate was a nightmare. I don't even know how to establish it, in a real cellular material. But maybe we don't need to. Maybe we can have some compromise, and that's what we would want to discuss with the community.
I think War and then Joachim.
Try to generate some synthetic data.
There's an entire synthetic data set that's identically matched to the phantom data, the same proteins in it, the same size things, the same ice thickness, on the portal that the machine learning community is using very heavily to train. But as you all know, synthetic data always works. You can always find the particles in a synthetic data. You can't necessarily find that in real data. And Chris and Richard and our group had a very interesting discussion yesterday about the very urgent need for good synthetic data, for synthetic data that models real biology. And I think that's an area we should be pursuing quite hard, in order to, solve some of these problems, and then, as Richard says, model them well so that we know what we should be going after and what is possible. We can't really do that yet.
Bridget, what are the criteria that go into the scoring?
It's all written up in the paper, and it's a slightly complicated criteria that the easy ones get weighted, differently than the harder ones. So easy meaning apoferritin, virus-like particles. The hard ones are beta-galactosidase, thyroglobulin, and you get scored heavily, you get negatively scored for missing a good one. You don't get so negatively scored, 16 times less worse, if you pick up something that we didn't, because we don't know, maybe it's a real one. We aren't sure because of the difficulty of, so it was a whole complicated thing that that was developed by somebody in the group to do that.
Do you apply conformational changes to the protein, or they're just fixed structures?
Yeah, we're not even close to that resolution. I think for the ribosomes we have tens of thousands of them, and for the virus-like particles we have quite a lot, and there's some symmetry. So we can get to decent resolutions with those. With the thyroglobulins, the beta-gal, the beta-am, we're at domain structure, so we're not seeing anything fancy or fussy. But for this, it's good enough we can just pick them, and then we'll take them further with other methods. Yeah, right now the picking is the problem. Thanks.
Thank you again, Bridget. Our final speaker in the session is David DeRosier, whose name came up in some previous presentations, and amongst other things, he was involved in performing the first single particle called reconstructions of icosahedral viruses.
So I wanted to begin to extend what Bridget has told you about the future. because I'm going to talk about cryo-electron tomography in 2075. The first thing you need, as Bob Glazer has said, is a quantum computer with AI software. If you've got AI software, it will tell you, it will pick what problems to solve and what systems to use. It then, of course, sends out requests to the appropriate companies to make the samples. And then it has the samples sent to the appropriate places like WAS S2C2 to be imaged. So the Cryo-ET movies are then inputted to the software. And of course, it appears in a paper in a new publication, Nature AI. And of course, there's no need, of course, the artificial intelligence, it selects a paper, edits a paper, reviews the paper, publishes a paper, and there's no need for you to read it because it will supply in your email a synopsis of what you should, the bottom line of the paper. So this is absolutely, I'm really a bit of an AI skeptic, machine learning, because I found out that the machine learned something, but I didn't. So, and I think Richard and some other people wrote a paper about the protein structure prediction, that it doesn't really end everything. Anyway, I wanted to back up a little bit with a slightly idiosyncratic, oh yes. So one of my favorite about making predictions is from Yogi Berra, who said it's tough to make predictions, especially about the future. I used to give old talks full of Yogi Berra quotes. People will remember. This is my idiosyncratic history of structural cell biology. And I think one of the first things was negative stain made a huge difference to how people could see things, because you could see molecular structures, albeit in projection. And then, of course, 3D reconstruction. And then the cold stage that people have mentioned, and of course then that Bob Glazer and Ken Taylor did, and then of course the glucose embedding, and then SPIDER, the first single particle software. These are the advances that I said wow to. So they're not obviously what Ava was a bit different. Then multivariate statistics, which is an extremely important addition to understanding images. Amorphous ice, and then of course cryo-electron tomography, subtomogram averaging, maximum likelihood, as mentioned before, and focused ion beam milling, and of course direct electron detectors and FEG guns. That doesn't count for all the advances in microscopy and computing and very important advances in software. Now this was a wow moment for me because Richard and Nigel, for the first time in these two-dimensional crystals embedded in glucose, you could see reflections. I don't know whether this is a pointer. Oh yeah, you could see reflections out that showed you were really looking at the structure of the protein and not at a hole left in by a negative stain surrounding the structure. So this really was a wow moment. And the problem is that you can't actually see in the image, you can't actually see the subunits in the raw image. And what made the work of Taylor and Glazer so important was that you could actually have contrast when you used ice. And this is, of course, the paper that people have referred to and so on. I won't spend time on it. It wasn't picked up particularly because of the fact that there was a worry about crystallization of the water, which can produce artifacts. So it sort of lay there for a while until Dubochet came along and introduced vitreous ice. At the time he published it, the paper was rejected initially because they were told in Nature that vitreous ice was impossible So the fact that they had evidence, no point paying attention. But it was an extremely important moment. And it's nearly the perfect place because it's as near to native as you can get. And of course, dose was important. At the time, people had said, only like so many electrons, 5 electrons per square angstrom or whatever. So the idea was you could take a picture like this, but you were sort of screwed if you wanted to do anything else until Wolfgang Baumeister came along and showed us you could actually make a tomogram of something embedded in ice. That was, again, another step that, maybe it's my naivete or ignorance, but it just seemed like amazing to me that this was possible. And following this, focused ion beam milling because most cells were too thick. And Mike Marco was the first one I know that showed that you could mill ice and still retain the vitreous nature. And I think that was an important, again, step. And of course, then the Baumeister lab and many people who are here helped improve it and make it into a technique that everyone is using today. And this really is, in fact, an important aspect. And this is from Elizabeth Villa's lab. And here we see a tomogram with slices through it, and it's been segmented. And you can see in the segmentation there are recognizable thin filaments and ribosomes and membranes and some pink stuff in the nucleus. But it brings me to the main point of my topic is how can you locate macromolecules of interest in the EM image? And I'm not going to go over all the methods. I'm going to talk about my involvement, which began, there are 3 methods I plan to talk about. It began in the 1990s. The first method was a gold tag. At the time, people were using thin sections, and then they'd take an antibody with a gold tag and bind it to the thin section, and you maybe got 1 or 2 or 3, and it seemed to me that that wasn't enough. And so we developed the idea to use a metallothionein, which is a metal-binding protein, to use it as a tag you could clone on to your molecule of interest, and then you would grow a gold cluster. So it seemed to me when you grow a gold cluster, steric hindrance becomes much less of a problem, so you should get much better labeling. And Chris Mercogliano was my last grad student before I retired, took up this project. Now, metallothionein is a small protein, 61 amino acids, molecular weight of 6.5 kilodaltons, 20 cysteines, and in this structure, you can see that it binds about 7 cadmium ions. So what Chris did was he cloned it onto maltose binding protein and labeled it with orothiol-malate, which will transfer the gold. And here are some early mass spec. And you can see if you have 1, you can get about 12 gold. If you have 2 metallothioneins in tandem, you can get 38. And in one case, he actually got 50. It's beginning to get up to the place where you could actually see gold in the EM quite easily. Cells really don't like gold salts. They really don't. And so people who have used this decided that wasn't really a particularly good direction, although I must say Christina Risco has shown that you can get labeling within maybe less than half an hour, and the cells don't show these cytotoxic effects for a couple of hours, so it may not be all over, but in any case, we had left it there, and Wanzhong He had gone on to really perfect the method for section cells in which you can fix the cell using, for instance, these cryo methods for fixing them, and then infuse in gold in the form of, for instance, gold chloride, and you can get labeling, and then here's a section from this. And you can see that there are a lot of examples of gold. This is a small membrane protein. So I think you shouldn't really run to cryo if the answer might be better obtained with an older methods than thin sectioning, where you can actually see the relative density and so on. So what's for the future of this? Well, I think gold labeling is a good way of doing things because it's small and it's visible. Now, Lindsay Young and Elizabeth Villa's lab has developed a method of gold tagging structures in live cells, not using metallothionein, that can be used with cryo-electron tomography. And I think Elizabeth is going to talk, she nods. And then, of course, there are other large clone tags that people can see that are proteins or nucleic acids that people can use to identify where in the cell their protein is. So that was method one. Method two was something I began when I retired. I had this idea that in 2005 that with a fluorescent tag, you can localize a fluorescent tag to better than 1 nanometer. People had done it. And so the question was, could we do this for a frozen hydrated grid? Could we localize something so you know when you looked at the EM, if you lined up your cryo super resolution image with your image in the electron microscope, you line them up, you could say my molecule is near this fluorescent tag. And of course, then in 2006 came a POM and STORM. And so the first thing we did to see if it could be, this was done at room temperature, but we wanted to see if you could do it in the cold. And the first thing we did in 2007 was to see whether PAGFP could be activated in the cold. Indeed, it could. And so we built a microscope around this idea with a high numerical aperture lens so we could collect the most photons. And this is sort of a diagram. Within less than half a millimeter, you can go from 25 degrees C to minus 190 degrees. Doesn't sound possible, but it is. And so we built a microscope. And now, when you have a frozen sample, everything is not straightforward because the dipoles of the fluorophores are fixed in space. And that means that the light arriving from them isn't as well behaved as if you have a tumbling dipole. So I wrote about it in this review, Where in the Cell is My Protein? The Difficulties and Alternatives. But we did build the microscope and we could isolate, we could show we could get super resolution. Localization was not great, 35 nanometers. This was just a trial. I think you could do better. But the postdoc, Mark Namani, went on, and I ceased my postdoc doing this work. But I think the important thing is we need to have a commercially available super resolution capability, both for cryo-light microscope and for FibSam. Maybe not so much as to identify where your molecule is, but where you want to mill a section. So if you have some unusual event, you want to mill the section there, you'd like to know where to do that. And this is, I think, doable, as people have said the physics doesn't argue against it. And I've described some of the difficulties in that paper of localizing them using super resolution. I think it's worth doing. The third thing I began more recently, I've been what I like to call fiddling with, is fill in the missing wedge. Now, the missing wedge is because you can only tilt between plus and minus 60. So there's this wedge where you don't collect any data. So if we could fill in the missing wedge, we could generate a tomogram that was closer to the truth and maybe thereby have a better tomogram. I was very skeptical. I lost the job because I was asked very early on, many years ago, what do you do about the missing wedge? And I said, it's missing. That was, and I think that ended the conversation. I've always been very skeptical about filling in the missing wedge. But I tested a method using a set of linear equations, actually, to recover the missing data from the observed data. And this was the same technique that Tony Crowther and I, with the help of Linda Amos and John Finch and Aaron Klug, developed by using a set of annuli. And the problem was, to get the first reconstruction, you had too many unknowns and not enough computer space to take a set of linear equations and invert them. But the thing about it is, when you calculate the normal matrix as you're doing it, inverting it, you can get a set of eigenvalues. And the eigenvalues, the inverse average, tells you how the error in the input is propagated into the data. So, of course, I decided I would do that for the missing wedge. So I've taken one circle here. And what I did first was, using the time-honored thing, I just made up some values for the coefficients. And I used those to generate the Fourier coefficients. These are the Bessel coefficients. I won't go into it. And you can add noise to it. So I can generate some model input. I can then use the model input to solve this set of equations, which looks the same, but it's not, because you're putting in your fake knowns, and they're asking for the, you're putting in your fake knowns and then asking to solve for the unknowns. And then you can use the solved unknowns to regenerate the output. To see how well you did, you can compare this to the input. So you can do that. And you can ask, what does the inverse eigenvalue, the average, look like? And if you have 5 orders input, which is not very many, you do quite well. And when you go up to 10, you can see you're already amplifying the noise by about 50%. And if you think about a thick section, to think about 10 orders is not really very much resolution. And what happens is that the errors don't show up everywhere. They'll pile up, guess where? Right in the middle of the missing wedge. So you'd say, if you looked at this, you'd say, oh, an error of 80 in a phase error is not bad, but this is an average. It means that for every one that was, let's say 80%, there was one at 180 degrees, there was one at 160 degrees. So this is really very bad news. And as you might expect, trying to solve this in 1D is kind of a bust. And the other thing I want to say about filling in the missing wedge is any nonlinear operation that you propagate on your data, even if it has absolutely no relevance to anything, will fill in the missing wedge. So filling in the missing wedge is not, just by amplitude, is not really a measure of how well you're doing. And I think given the rate at which errors are propagated in, really leaves me very skeptical. I'm willing to be convinced, but it may be that some of these algorithms are just making bumpier things bumpier. Anyway, so I thought maybe looking at this in 2D might be better. So if you do it in 2D, you can enforce that the lamella is only so thick. And also, if you remember that segmented tomogram, you have a lot of known structures. So the idea is, if you know the structures, they're no longer unknowns. You can replace those structures with the known structures. Now you've reduced the number of unknowns. Can you, in fact, fill in the missing density? I fiddle with it. Anybody is welcome to join me in fiddling. It's an idea. I don't know whether it'll work or not, but it seems to me the more straightforward way of trying to fill in the missing wedge where you actually know what you're doing. Finally, I'd like to end with a quote from Bob Glazer, who said, "We overpredict what will happen in the short term and underpredict what will happen in the long term." And having started with negative stain and thinking we'd never do any better than molecular resolution, I must say I am stunned by how well the community has done. Thank you.
Thank you, David, for a fantastic talk. Questions? I did have so there's a question from Ken, actually. Ken.
So if you use, try to fill in the missing wedge with, say, you've identified ribosome, you fill in the missing wedge with ribosome information that, that's not getting, I don't see how that's getting you anywhere. It's not telling you anything about other stuff that you don't know.
No, it does, because what it does, it reduces the number of unknowns. So when you come to solve the equations, you'll now get a better tomogram. So you start off with your initial tomogram, you take all the structures you can recognize, and you can subtract them in reciprocal space effectively, and you're left with fewer unknowns. So now the problem becomes better conditioned. So the answer means that the noise from the input data is in fact better handled because you have fewer unknowns. So you're not looking to solve the ribosomes, you're looking for something smaller that you don't see that looks like noise in your tomogram. You want to make those densities better. That was the idea. I'm sorry I didn't make that clear.
Well, there's another collection of constraints and limiting the data, certainly in Fourier space, where you could apply the, I think you've heard of it, the Crowther criterion, and say that since the data that we have from -60 to +60 has a limitation on its own resolution, that you're not allowed to trust anything beyond that resolution, which for a tomogram is 50 or 60 angstrom resolution given that thickness. You'd say, don't even try to fill in the fill in the missing wedge beyond that resolution, and that's another constraint.
Yes, exactly. So there's only a certain, in reciprocal space, there's only a certain resolution within that reciprocal space you need to worry about. In the case of Bessel functions, it's how big are the Bessel functions you have to worry about, right?
Okay. Well, let's thank David again.